Okay, let's try this again. We're going to talk about how you mobilize phagocytes to join the fight against pathogen, an invader. So the first thing you do is leukocytosis. You're saying we got to get some white blood cells. We're going to release neutrophils that we're developing in the bone marrow. We're going to make them grow up and release them really fast. That's because the cells that are injured or at the site of infection, they release leukocytosis inducing factor. It's just a chemical that says, hey, we need white blood cells, we need neutrophils, send them now. The next thing, you're going to have margination. The endothelial cells in the wall of the capillary, we're talking about the wall of the capillary itself, they start sticking out. Remember, they're normally smooth, slippery, everything slides by. They start putting little projections, little cell adhesion molecules out that will try to grab onto the neutrophils and slow them down, stop them. This will cause the neutrophils to stick to the wall and roll along slowly until they finally will flatten out and squeeze through the clefts between the cells. We call that process diapedesis. Remember that word, it's a good word to know. Chemotaxis. Taxis means movement in a direction, and chemo is what they're doing. They're following chemicals. Basically, they're sniffing their way towards the injured area. So neutrophils are going to get there first, which makes sense because they're the most common. But they're probably going to be overwhelmed and getting their butts kicked and dying in the process of the battle with the bacteria. About 12 hours later, the big eaters, the macrophages, are going to show up. Actually, they're going to show up as monocytes that will then grow into macrophages. But they replace the dying neutrophils, and they're going to stay afterwards to help clean up the pieces of all the damaged cells. So here we have an injury, and we got some bacteria, and the bacteria are invading the tissues. And let's go through step by step. Leukocytosis, we deliver our white blood cells to the area. Margination, the margin, the edge of the capillary gets sticky, so the white blood cells are sticking to it. Diapedesis, they flatten out and squeeze through clefts between the endothelial cells. Chemotaxis, they are going to follow the smell of the damage, follow chemicals that are being released from the injury, from the injured cells, and go to the wound, and there they will discover bacteria that need some whooping. All right, so here we have events of acute inflammation. It's a flow chart. It's pretty basic. It goes over the things that we talked about so far. Innate defenses, internal defenses. The inflammation response is right here. This is a good chart if the things we said are going too fast. You can use this to refresh your memory. You can use this to quiz each other. Good method. But we're going to move on, which means we're going to then move into the adaptive defenses. Anyway, so homeostatic imbalance, you're going to have a bunch of dead neutrophils at the wound, and that means you're going to have pus. Pus is a yellowish, whitish, it's a bunch of dead neutrophils, also dead cells and tissues from the wound, and living or dead pathogens. You might have the body try to contain the damage to one area by laying down a bunch of collagen fibers and making a sac. That would be what we call an abscess. And an abscess full of pus, you might have to slit it open and drain it. Disgusting. So some bacteria are hard to kill by macrophages, things like tuberculosis, and they're going to remain alive inside. We could get these tumor-like growths called granulosas or granulosums, and It's an area of infected macrophages surrounded by uninfected macrophages surrounded by a capsule. The bacteria could stay in there inactive forever, or if someone's immunity is compromised, they could break free, become active, and start up the disease. So, antimicrobial proteins are going to be used. They could attack the microbe directly, like attack a bacteria directly, or they could make it hard for it to reproduce. The two we're going to talk about the most are the interferons, which interfere with it, obviously, and the complement proteins. So interferons are a family of proteins that if a cell is infected, it can send out interferons to warn its neighbors, I'm under attack. They're coming for you to get ready to fight. So interferons will go into a neighboring cell. They will tell that cell to make proteins that will block viral reproduction and break down viral RNA. We have type A and type B, I should say type alpha, type beta interferons do that. We also have another option, which is the alpha and beta have a separate path of activity where they can activate natural killer cells. Remember those lymphocytes. So the gamma is also called immune interferon, and this one is made by lymphocytes. It wakes up lots and lots of things. It activates macrophages. Interferons can wake up the natural killer cells, the macrophages, so they indirectly can be used to fight cancer. We've talked about that before. Artificial interferons are used to treat hepatitis, genital warts, and multiple sclerosis. So we can make our own. Anyway, so here we have an infected cell. We have it getting infected. That's what you see at 1 right here. That's a virus injecting its nucleic acid core. The idea being that the cell will then copy and make new virons and release them to infect the next cell. That's what it should go, but we're going to show the power of the interferon. All right, so when the virus injects and is detected, the interferon genes are activated. And when the interferon genes are activated, the cell makes interferon molecules, which are released, go to the next cell. Those activate the cell to make, how did they word this? Binding stimulates the cell to turn on antiviral proteins. And these antiviral proteins will be there ready for when the virus tries to invade to stop it from being able to reproduce. So antiviral proteins block viral reproduction. Yay. Okay, so that's what interferons do. Complements are cooler. Those are the ones I talked about that rip holes in the bacteria. So complement system, about 20 different proteins. They're circulating in the blood in inactive form. They can directly destroy foreign substances. When they're activated, they will make inflammation stronger, but they will also try to directly destroy the bacteria. Complements help both the innate and the adaptive defenses. And the complement protein can be activated by antibodies that will bind to the organism. Remember, that's opsonization. And then bind to the complement proteins, activating them. This double binding would be called complement fixation. Once that happens, complement proteins are activated. There's a cascade, just like blood clotting, there's a cascade of activity. You could also have the lectin pathway. The innate system will make chemicals called lectins that recognize foreign invaders. When a lectin binds to antigens on the invading cell, it can also bind to a complement and activate the complement. The complement cascade can be activated spontaneously when certain complements bind to a foreign invader. Like one of them binds and they tell the others, hey, activate. The lack of inhibitors on a microorganism surface allows the process to proceed. So we normally have inhibitors that tell the complements, oh, chill, we don't need that. But each path involves the activation of proteins in an orderly sequence. This is the cascade they're talking about. Each pathway converges on C3. So all three of those ways of doing it, you're going to end up with this C3 molecule. C3 is going to break into C3A, C3B. When it splits, this will be the end of all of the pathways. They all do the same thing at that point. They enhance inflammation. They promote phagocytosis, meaning the neutrophils the macrophage is eating. And here's the fun one, and cause cell lysis. When the C3 binds to the target cell, it's going to cause a membrane attack complex to form or to come. And it says membrane attack complex forms, stabilizes a hole in the microbe, causing an influx of water, lyses the microbe. A picture's worth 1,000 words here. You have a bunch of these little C3s that come together, and you have a C3 with a C5b, a C6, C7, C8, C9. That forms a rod, and that rod goes into the membrane. It says the one next to it, the one next to it, until you have a hole through the middle of the membrane. Think like a tunnel, and water can rush in, and the bacteria will swell. It'll explode as it fills with water. All right, so C3b will also do opsonization. Remember, that means to make it tasty. That means it makes the phagocytes more likely to eat the thing. It will cause other complement proteins to amplify the inflammation. The mast cells and basophils are going to release histamine. That's going to make your vasodilation of the blood vessels, make the blood vessels more permeable. The complement protein is going to be one of the things that attracts the neutrophils. Remember, we talked about chemotaxis. One of the things they're sniffing for is this C3A. Anyway, so we've got that. Now let's talk about fever. Why would we want a fever? Because if the body temperature is too high, it can interfere with the bacteria's ability to reproduce itself. Leukocytes, macrophages, they're exposed to foreign substances, and when as a response, they're going to create pyrogens, pyro like fire, gen to generate. The leukocytes and macrophages, when they find the bad thing, like, oh, we need a fever to help control this, and they release chemicals that call for fire, chemicals that call for fever. The pyrogens are going to affect the hypothalamus, raise the body temperature. And if you have a moderate fever, the liver and the spleen are going to grab all the iron and the zinc that they can out of the blood. That means it's not going to be available to the bacteria to reproduce. It's going to increase your metabolic rate, which means you have more energy available to grow new tissues and repair the wounds. Here you go. Here is table 21.3. That's the second line of defense. We still haven't gotten to the adaptive things. Okay, so that will be the next PowerPoint, I assume. Hold on. I'm going to pause this for a moment. Okay, and now we're going to start on the part B PowerPoint. We're going to do the adaptive stuff. We've been through the innate, the stuff that's not specific to the invader. You remember that was the physical barriers, and then the phagocytes. The adaptive system is more powerful, but it takes longer to ramp up. It will destroy most pathogens, most abnormal cells in the body. Um, we're going to amplify the immune response. We're going to do lots of the complements. Um, the problem, of course, with it is that it takes time. You have to have initial exposure and then build a response. Say, to make antibodies, you have to divide one of the memory B cells, and then one of the two daughter cells you get has to grow up into a plasma cell, and yeah, it takes some time. Um, but this is specific. It recognizes specific types of antigens that it's been exposed to. It is systemic, meaning those antibodies go everywhere. And it has memory. Remember, there are memory B cells and memory T cells. We tend to talk more about the memory B cells because those are the ones that make the plasma cells, which are antibody factories. And it gets faster every time you expose it to the pathogen. Like the first time, it takes a while to learn how to fight. The second time, it comes on much faster, and third and fourth. That's why booster shots, to make sure that you're ready to go, make sure that the memory cells are doing their job. Let's see. So, two types of adaptive. We have the humoral. That means the body's humors. That's an old-fashioned way of talking about the body fluids. Antibodies float around in the fluids. Quick recap to remind you again, an antibody is part of your body's immune system. It's a little hunter-seeker probe that looks for bad things. Your body makes these antibodies. The plasma cells make these antibodies and release them to look for things that don't belong in your body. Anyway, so humoral, the body humors, the body fluids, and based mostly on antibodies floating around. We also have cellular based immunity, cell mediated immunity. We're about to jump into both of these. So, humoral immunity, this is caused by antibodies produced by lymphocytes. Will be very specific because I've already said it. Produced by plasma cells, which are a daughter of a B lymphocyte, a memory B cell. Antibodies float around in the fluids. They will bind to the target cell temporarily, and the target cell being the invader, the pathogen, call it a bacteria, say. They can temporarily inactivate some of them, but it says can mark for destruction by phagocytes or complements. Remember, we said that you can bind to things and do opsonization to make it tasty. You attract the phagocytes to make them grab on and give them more structure to grab onto. Also, not mentioned here yet, but many antibodies, many immunoglobulins can grab on to more than one pathogen. So it's hard for bacteria to swim away and evade a white blood cell if you have two or three of them hooked together by the same antibody, all trying to swim off and going in different directions. Anyway, but yeah, then inactivate the invader. It can mark it for destruction by phagocytic cells, neutrophils, macrophages, or complement proteins. Rip a hole in the membrane. Humoral immunity has extracellular targets. It's looking at the outside of the cell. It's looking at glycoproteins on the surface most of the time. Cellular immunity, this is lymphocytes that are going to attack. They can do it directly by killing infected cells. They can do it indirectly by releasing chemicals that ramp up the inflammation, or activate other lymphocytes, other macrophages. An example of activating others, helper T cells, which we've mentioned before, are a form of T lymphocyte. They're kind of like the generals telling other cells, like telling the soldier, you need to go do this, you need to go do that, you need to go over there and do that. That's a helper T cell. If a helper T cell gets infected, it's really bad because that's the one that should be running the defense. That's how HIV happens to work. But we'll talk more about that later. Anyway, cellular immunity has cellular targets. You're attacking cells. So let's go through the antigens, which is part of the humoral immunity. Okay, antigens can mobilize adaptive defenses, provoke an immune response. Remember, an antigen is anything that generates an immune response. Don't confuse antigens and antibodies. The antibody is part of your body. The antigen is something that does not belong in your body. It is something that your body wants to fight. So antigens can cause an immune response. And they are the targets that we are honing in on with our adaptive immunity. Most of them are large, complex molecules that shouldn't be in the body. And what are the characteristics? It could be a complete antigen or a hapten, which is a partial antigen. It could have antigenic determinants. It could be a self-antigen. So going through each of those, an antigen can be complete or incomplete. That means they have two important functional properties. Immunogenicity, which means it can cause us to make a bunch of lymphocytes as an immune response. Or reactivity, the ability to react with an activated lymphocyte or an antibody that was released from an immunological reaction. So basically, what are examples of antigens? Foreign proteins, polysaccharides, lipids, nucleic acids that don't appear to belong. We see these on pollen, we see these on bacteria. Great. All right, incomplete antigens, some people call them haptens. Think of the H as remind you of half of an antigen. They are too small to be seen. They are not immunogenic by themselves. This could be some hormones, some peptides, nucleotides. But if the hapten attaches to the body's own proteins, the complex, the combination, could be seen as foreign and not belonging. All right. This could cause the immune system to attack self proteins as well as haptens. Examples would be poison ivy, animal dander, some detergents, some cosmetics. Antigenic determinants. The determinant is the part of the antigen that will be grabbed onto by an antibody, by a lymphocyte receptor, or sorry, by a lymphocyte. Most naturally occurring antigens have numerous antigenic determinants, meaning most antigens, you have this little antigen here. This part of it could be something that is a determinant. This part, this part, this part. There are lots of spots on that because the whole thing is foreign and weird and doesn't belong. There are lots of places on it, little tiny spots. You look like, yeah, that doesn't look like one of our cells. Those are what the determinants are. Large, chemically simple molecules like plastics have no immunogenicity, don't cause immune responses. So here we have an antigen, and this antigen has several determinants in different shapes, several different molecule things, different shapes. And different antibodies can bind to those antigens. Notice that antibody C matches this determinant. Antibody B matches this determinant right here. It matches this determinant here. Antibody A matches this determinant here. What we're saying is that when antigen that has lots of determinants, there are lots of different antibodies that might be able to bind to it, but they only bind in one way, one specific connection. It's kind of the same as an enzyme and the active and the substrate. The active site of the enzyme is specifically shaped for the substrate. Same deal. The antigen has this determinant, and the antibody that will recognize it as being the bad thing, it will bind to that one determinant, not the 2 or 3 others, that one determinant. So self-antigens. All of the cells are covered, all cells in us are covered with a variety of proteins on the surface. They're not antigenic to self, but could be antigenic to others in transfusions or grafts. What they're talking about is things like your ABO blood complex on your surface of your red blood cells, the A, B, or O variant. They determine your blood type, whether you're type A, type B, type AB, type O. Those little proteins would be considered to be self-antigens, okay? One important self proteins are glycoproteins, major histocompatibility proteins, unique to each individual. That's how, if you get an organ transplant, your body can say that doesn't belong, or you get a blood transfusion, your body can say that doesn't belong. We have to get someone who is a good match that has as little difference from you as possible. These self antigens have a groove that could hold a piece of they have a groove that could hold a self antigen or a foreign antigen. T lymphocytes, the ones from the thymus, can only recognize an antigen presented on a major histocompatibility complex protein. Yeah, they don't just see one random. It is brought to them by the complex. All right, so we've got 3 basic types of cells in the adaptive immune system, and we're still talking humoral here because we're talking antigens and antibodies. 2 lymphocytes, B and T cells. The B ones are the humoral and the T ones are the cellular. That means the B ones are involved with antibodies, antigen. The T ones are involved with directly attacking cells. Antigen presenting cells are the third thing. And those are the ones that will bring a piece or usually a piece, but bring a potential antigen to one of the 2 lymphocyte types and say, look at this thing, it seems bad, I think you should fight it. Antigen presenting cells don't respond to specific antigens, but they are very important in showing them to the 2 types of lymphocytes. So lymphocyte development, maturation, activation. Remember, never let monkeys eat bananas. All of so lymphocytes are the second most common. Never let monkeys eat bananas. Lymphocytes are the second most common type of white blood cell, and they're going to start life being formed in the bone marrow, like all white blood cells do. And then they're going to mature. They're going to grow up in a primary lymphoid organ. The B ones are going to stay in the bone. The T ones are going to go to the thymus. Okay. And during this process, they are going to learn what is normal and what needs to be fought. And fancier saying, they'll become immunocompetent, competent or capable of doing immune things. The lymphocytes have to be able to recognize one and only one antigen. So B or T cells display one unique antigen receptor on the surface when mature, so they can only bind to one specific antigen. And they need to be self-tolerant. We don't want them attacking normal cells in your body. We don't want them causing autoimmune disorders. Okay, so they have to develop competence and self-tolerance. Competence meaning they have one thing they know they're going to attack, and self-tolerance meaning they don't attack other things that they shouldn't. All right. So 3, seeding of lymphoid organ circulation. B and T cells that haven't seen an antigen yet are what we call naive. They are sent to secondary organs. That could be the spleen, could be a lymph node, but they leave the bone marrow or the thymus and they go to some other place. In that other place, there's a real good chance that they will find an antigen. So immunocompetent but still naive lymphocytes leave thymus and bone marrow. They will seed secondary organs as they circulate through the blood and the lymph. I want to emphasize that B ones will tend to be more stationary once they get to a spot, a secondary organ. The T ones tend to travel. All right. I should have said are more likely to travel. Anyway, naive lymphocytes will find an antigen, will encounter an antigen, and it will cause the lymphocyte to develop, to change. It will differentiate into an active lymphocyte by binding to a specific antigen. We call that clonal selection. And if the correct signals are present, it will complete differentiation, will become an active cell. All right. So antigen encounter and activation. When a lymphocyte's antigen receptors bind to its antigen, that lymphocyte can be activated. Great. Okay. And proliferation. That's what you see at the bottom here, where one cell will become many cells. Once it's selected and activated, the lymphocyte proliferates. It forms an army of copies of itself, of clones. Yeah, I know there's a Star Wars joke about the Clone Wars embedded in there, just begging to be asked, but I don't know Star Wars well enough to make the joke. Anyway, but yeah, you make an army of clones. And those are the effector cells, the ones that are going to do the fighting. A few of them are going to stay back as memory cells. And remember, there are T memory cells, there are B memory cells. They're able to respond to the same, they remember the previous infection, and if it happens again, they can respond faster. These B, these T, and these effector cells, effector T cells are always in circulation. Okay, yeah, so we got that. Genes, not antigens, determine what foreign substances the immune system can recognize. There are a bunch of different immune cell receptors, and they're the result of acquired genetic knowledge. There are 25,000 different gene codes for 1 billion different types of lymphocyte antigen receptors. A lot of variety, obviously. Those segments get shuffled around, making new combinations. Great. You already know that T cells grow up in the thymus, where they're tested. They have positive and negative selection. Positive selection, we look for T cells capable of recognizing the major histocompatibility complex proteins. Those that can't, we tell them to kill themselves, apoptosis. Like, if you can't recognize a threat, you must die. Then we do negative selection. And if a T cell binds to your own cells that are normal, we don't want that around, that's going to be told to kill yourself. Apoptosis is when a cell kills itself. So that's called clonal deletion, and that ensures self-tolerance. Okay. So T cells, positive selection, they have to be able to recognize something as bad, and they have to not think things are bad when they are normal. Great. Antigen presenting cells. They are surrounded, they grab the antigen, they break it apart, and they show pieces of it to T cells. Okay. Different types. We have dendritic cells, we have macrophages, we have B cells. All right. So dendritic cells are found in connective tissue in the outer layer of your skin. They let you know if something's trying to cross the boundary. These dendritic cells can phagocytize pathogens that get into the tissues. They can go into the lymph areas, lymph nodes, through the lymph vessels, and present those antigens or antigen fragments to the T cells in the lymph node. These dendritic cells are the most effective antigen presenter that we know about, and they are the key link between adaptive and innate immunities. So good to know that dendritic cells are the most effective antigen presenter. And there's a dendritic cell. You may be, that looks crazy. Yeah, but do you remember when we talked about the nervous system and we talked about the cell body and the axon, but on the cell body, there are all those extensions called dendrites? That's probably where dendritic cells get their name, because kind of the same. All right, so antigen presenting cells, you could have the dendritic cells. You could also have macrophages, grown up monocytes, white blood cells. They're capable of talking to other white blood cells and saying, hey, this thing that I've been eating, this thing that I've been fighting, yeah, you should get ready. This is bad stuff. Well, you should get a whole army together to fight. So macrophages are found all over in connective tissues, lymphoid organs. They will show antigens to T cells that'll activate the T cell, but it will also activate the macrophage. In other words, macrophage, sir, I have to show you the thing, the antigen or the fragment of the antigen. And when the general, the helper T cell, gives a positive response, the macrophage is excited and it goes out and it gets better at fighting. It becomes, in the words of the textbook here, a voracious phagocytic killer. And it also triggers powerful immune responses and lets you recruit additional defenses. I wish they itemized them there, but anyway, we'll assume that's coming up. So the B lymphocytes, they don't activate naive T cells. They don't do that. But they do present antigens to helper T cells. Remember I said those are like the generals. They will present antigens to helper T cells to assist in their own activation. Oh, really bad sentence. Horrible use of there. Anyway, so here is the table clarifying the difference in B lymphocytes and T lymphocytes. It is table 21.4. I would say this is important to know. Let's see, types of immune response. Yeah, you should know that. Antibody secretion. Yeah, you should know that. Primary targets. Yeah. Origin. Yeah. Maturation. Yeah. Effector cells. Probably that too. Yeah. Unfortunately, I have to tell you this table, know the table. All right, so humoral immune response. Remember, this is a B cell thing. Oh, hold on. Hey, babe, hold on. Stop the share. Click stop the share. Okay, so humoral immune response. When a B cell encounters an antigen, this starts the humoral immunity up. Our goal is that we are going to make antibodies against that antigen. So this is the process how. All right. So B cells get activated when an antigen binds to a receptor on the surface and cross-links with it. This triggers receptor-mediated endocytosis. Fancy way of saying that the B cell will then pull the thing inside of itself. And it's pulling the antigen and the receptor that it's stuck to inside of the B cell. This leads to proliferation, meaning the creation of lots and lots of B cells, and differentiation, which means that some of those B cells are going to change into effector cells, meaning plasma cells. Okay, moving on. Most of these clone cells become plasma cells. Those are antibody factories. Some make specific antibodies at a rate of 2,000 molecules a second and do that for 4 or 5 days until they wear out and die. Just run full blast to create lots of these antibodies. What are the antibodies doing? They're floating around in the blood looking for whatever has that antigen, whatever is invading, whatever doesn't belong, they will bind to it, and then they will call the white blood cells to come over and get it. So antibodies circulate in the blood or lymph, binding to free antigens, marking them for destruction by innate or adaptive immune systems. Okay. Here is a picture. Antibodies are out there and an antigen binds to them. This is one of two. Yeah, one of two. Okay, so antigen binding to a receptor, specific B lymphocyte, talking a memory cell. And I'm gonna move this so I can read it. There we go. Antigen binding to a receptor on the specific B cell lymphocyte. B lymphocytes with non-complementary receptors remain inactive. So this little star-shaped, asterisk-shaped antibody, antigen will fit this antibody. It will not fit, this plasma cell. It will not fit that one or that one because they're slightly different shapes. The activated ones will be activated and they'll clone. Okay. And when they clone, most of them become plasma cells that create more of those antibodies while floating around. But some of them go on to be memory B cells that replace the activated memory B cell. So you can remember for the third or fourth time you're exposed to this, you'll always have some around. All right, subsequent change to the same antigen results in more rapid response. Great. We've talked about that before too. All right. Clone cells that do not become plasma cells will become memory cells. They will provide an immunological immunity, mount an immediate response to future exposure to the same antigen. Again, we've said that. All right, so the secondary response, another time later on when you're exposed to the same thing, when this happens, you will make lots and lots and lots of plasma cells, way more, and lots more memory cells because, oh, this happens more than one time. We need to remember it even more than we were. Super bad. And that means you can have faster responses to the second and third exposure. So the primary immune response, cell proliferation and differentiation, it's going to take 3 to 6 days to happen. And your peak antibody level is going to be 10 days later, and then the antibodies will decline. But the next time you're exposed, because of the memory cells, your body will respond within hours, not days, and your antibody levels will peak in 2 to 3 days instead of 10 days later. The antibodies will bind tighter, and the antibody levels will remain high for weeks to months. You basically take the immune response and you turn it up to 11. Anyway, so here we have first exposure to antigen A, you have a response. Second exposure, way stronger. Now, antibodies to B are, if you've never seen B before, even though you have a super strong reaction to A going on, you're going to have the same kind of slow first exposure to B. But the second time you're exposed to B, it'll be really strong. All right, so active and passive humoral immunity. Active humoral immunity is when B cells find an antigen and they produce antibodies against it. There are two types. There's naturally acquired. This would be like you're exposed to bacteria or an infection. And there's artificial. And in artificial, a good example would be a response to a vaccine, where they give you weakened or partial pathogens. The idea is that you want to provide some antigenic determinants that will cause an immune reaction and then you won't have the symptoms that come with the primary response. You'll just have the development of the memory cells and be ready for exposure. Now, passive humoral immunity will occur when ready-made antibodies are introduced into the body. The B cells are not challenged by antigens. Okay, this is the example that they're talking about right there. Naturally acquired. You've heard people say, "Oh, well, mama's milk is really good for the baby." No, mom's milk, especially the colostrum at the beginning of pregnancy, uh, sorry, right before nursing starts, not beginning of pregnancy, near the end of pregnancy. That colostrum is going to be full of antibodies. That milk is going to be full of antibodies from the mom. The mom, before the baby is able to make their own antibodies, the mom will supply some antibodies to the baby through her blood, through her milk, and that will give the baby a start in life. Artificially acquired the same way. So what I want to focus here on the words are passive and active. When you see active, okay, these are both humoral because they both involve moving around in the body fluid. So humoral immunity is what's floating around there, and we're talking the antibodies. Okay, so humoral is antibodies. Sorry, well, humoral is body fluids, and what's floating around in those fluids are antibodies. Active is anything that causes you to make your own antibodies. All right. You make your own antibodies under active immunity. Passive is you are given antibodies, so you don't have to wait for your body to develop the ability to make antibodies. So where they're saying artificially acquired injection of serum like gamma globulin protects immediately but ends when the body is degraded in the body. They're not giving you an example here, but I'll give you two. Oh, doc, I was bitten by a bat or a rodent, and I don't know if it, well, here, if you're worried about rabies, we're going to give you a bunch of shots. What are those shots? They're giving you antibodies. Your body's not making them, but they're giving you antibodies to protect against any virus of rabies that might have gotten into you. So now they'll probably give you also at the same time shots so your body will learn to make its own antibodies. That would be this artificial immunity. They're not going to wait for the natural. The natural would occur if you were exposed to the bite, but you by the time it works, you might have a disease. So they're going to be doing this artificial passive, giving you antibodies as if you did. And I don't know for sure, but I think they'll probably also give you the artificial active, meaning we'll give you some shots of dead virus, partial virus, so you definitely will develop your own antibodies for the future. But yeah, when you see passive, that means we're providing you with the antibodies, you're not making them. All right, here we go. So natural, easy to understand. Artificial, so natural, active, easy to understand, you got exposed. Artificial, active, easy to understand, you got a vaccine that will cause you to create antibodies. Natural, passive, mama's milk. The one that's hard for people to understand is passive, because, and the reason it's hard is we still will use the term vaccine, but we don't say, I'll give you an example where we don't say it. Snake bite, okay? If somebody gets a snake bite, we call it anti-venom, okay? But what it is antibodies that will bind to that venom, stop it from working, and then attract cells that will remove it from the body, destroy it. So anyway, moving on. So another name for antibodies, I've been saying antibodies all along, we could call them immunoglobins. They're made by plasma cells. You already know the plasma cells are daughter cells of memory B cells. And the immunoglobins make up what we call the gamma globulin in the blood. Okay, what's all this glob, glob, glob? They're kind of rounded proteins in some portions, and so that's where we get the globe. Anyway, the antibodies bind specifically with an antigen that was detected by the B cells, and we group them into 5 different types of immunoglobins. The basic structure of a immunoglobin, of an antibody, they're T or B shaped, 4 looping peptide chains hooked together by disulfide bridges. Nice strong covalent bonds there. These 4 chains are 2 heavy chains, 2 light chains. The variable regions at the end of each arm combine to form 2 identical antigen binding sites. What they're saying, here's a picture, here's another picture. When you look at these, if you think of it as a Y-shaped molecule, the end is the antigen binding site. The ends of the Y sticking up, like antigen binding site over here, antigen binding site over here. Okay. The stems in the middle, that'd be where my thumbs are here. Those stems are what let us put it in one of those 5 classes. Those are considered to be the effector region of the antibody. Several common functions in all antibodies dictating how cells or chemicals that the antibodies combine to. how antibody class functions eliminate antigens. Some of them fix complement proteins, like they will find an antigen and they will grab onto it, and then they'll grab a passing complement protein and stick it onto it. Some are just floating around the blood. Some are found in other body secretions, like your tears, your saliva. Um, all right, so we said there were 5 classes: IgA, IgD, IgG, IgE, IgM. Great. IgM, the M should remind us of massive. This is bigger than the others. It's the first one we release when there's a reaction. And it says potent agglutinating agent. What does that mean? That means that it's going to grab onto multiple antigens and clump them together. Like, say the wrong blood got put in your body, antibodies will grab onto this red blood cell and this red blood cell and pull them together, and maybe that red blood cell and pull them together, and you'll have a little cluster that doesn't move as quickly through the body, doesn't move as easily because it's too big. And that makes it easier for a white blood cell to track them down and eat them. Anyway, readily fixes and activates complement proteins. Great. So also puts those things onto stuff that can pop a membrane. IgA, monomer or dimer. This is released in mucus and other secretions, saliva probably. This helps prevent things from getting in the first place, stops them from entering. All right, so here we have pictures. The IgM is massive, and it's got 5 of these antibodies attached to each other floating around. The IgA is a dimer, it's got 2. Now D, G, and E, if we look right here, these are smaller. These we have single ones, so they're monomers, mono for 1. IgD attaches to the surface of B cells. It's a B cell receptor. G, it's a monomer. This is what the majority of the antibodies are. 75 to 85% of the antibodies in blood plasma. This is a secondary or a late response. These things can cross the placental barrier. They're the things that mom can donate to baby, and that would be good in many cases. Of course, they're also the things that can cause problems with Rh factor in babies and mothers, and that would be a bad thing. All right, so monomers activate some allergies and parasitic infections. This causes mast cells and basophils to release histamine, which you remember that's going to cause your inflammation response to ramp up and get stronger. That's not just going to make your nose run, it's going to make your blood vessels super permeable and cause them to vasodilate. All right, so there are the classes. Basically, table 21.5, look over that. I may pull 1 or 2 questions from it. Now, plasma B cells can switch from making one class of antibodies to another, but they will still be making antibodies against the same antigen. IgM is released in the early response, but the plasma cell can switch to IgG for the secondary response. Almost all secondary responses are IgG. You can see that is why 75 to 85% of the antibodies floating in the plasma. So what we're saying is, when we make a plasma cell, when a memory B cell becomes another memory B cell and a plasma cell, the plasma cell is going to start out making these big 5-prong, this pentamer, this IgM, and the massive one. And then later on, as the response builds up, You got to make these smaller ones that float around easier, that fit into tighter places and stuff. These IgGs. You could also switch from IgM to IgA or IgE. You just don't do it nearly as much. IgG is your main tool. All right, so antibodies don't destroy the antigens. They will inactivate the antigen. They'll tag the antigen. When they stick to it, they form a complex. And here are mechanisms used by the antibodies. Neutralize the antigen. Agglutination, which is essentially sticking, clumping them up, clump the antigens together. Precipitation, making the antigens fall out of solution. And complement fixation, sticking complement proteins onto the antigen or the thing the antigen is part of. So neutralization is simple, but it's very effective. Antibodies block specific sites on the virus or the bacterial exotoxin. They prevent the antigens from binding to receptors on tissue cells. And antigen antibody complex undergoes phagocytosis. So let me summarize here. What they're saying is, say there is a bacteria or a virus that's trying to get into cells. All of the molecules that it uses as hands to grab onto your tissue will gum those up. We will release antibodies that will grab onto the hands, the molecules they're sticking out, and clog them up, so the virus, the bacteria is not able to grab onto your tissue cell and get in. Great. And when they bind, they then they call to the white blood cells, hey, come over here, we're doing things. In other words, they promote chemotaxis. They release a trail of chemicals that can be smelled, that the white blood cells can follow. Okay, agglutination. Find the same determinant on two different antigen or on two different antigens at same time. Each antibody has two arms containing a variable region of binding to one antigen. This is the example I talked about with mismatched blood cells, where you grab a bunch of them together. This lets the antigen antibody complex cross-link into large lattice of clumps. Yeah, great. Basically, you cause the blood to clot. The mismatched blood, the blood that shouldn't be in your body, everybody's like, oh, I'm gonna stop that from floating around. And you form little clumps or clots that are slower to move around and easier for macrophages to catch. All right, precipitation. Soluble molecules instead of cells, cross-linked into complexes, so they're big and they fall out of solution. Venom, I talked about snake venom, and you get the anti-venom shot. What they're actually giving you is passive immunity. They're giving you immunoglobulins. This is a good example of that. Complement fixation. This would be, it won't work on a virus because the virus doesn't have a nice plasma membrane to rip apart. But this is the main defense against bacteria, mismatched red blood cells. Several antibodies bound together on the same antigen. Their stems get aligned, and they trigger a complement which rips the cell in half. Remember, the complement is a bunch of these little rods of protein or C-shaped pieces of protein. They're chunk, 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 and then they spread out, leaving a hole in the middle of the membrane. And what happens? Water rushes in, and the bacteria or the red blood cell swells, pop. Okay. So, mechanisms. We have neutralization that stops this virus from grabbing onto the tissue. We have agglutination, We are holding a clump of the red blood cells together. Precipitation, we're binding the nasty things and then falling out of solution. That would probably be, like I said, the antivenom. Antivenom being the antibodies here, venom being the little molecules they're grabbing onto. Complement activation. Here you bind to the cell and you to the outer surface of the cell. You take complement proteins, you stick them chung 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 chung, and now you've got a hole. Okay. And the complement activation, that just all by itself will kill the cell. Back up. All right, you can see right here that complement activation would also cause phagocytosis and enhance inflammation. All of the other methods, neutralization, agglutination, precipitation, they're going to cause phagocytes to come and eat them. They're going to cause the inflammation response. So, all right. So say you have a parasitic worm attack, Ascaris, that's a roundworm, something like a pig whipworm. Cystoma, they have different strategies. Worms that are too big for precipitation or lysis, agglutination, you still got to stop them. The IgE antibodies, immunoglobulin E, and let's back up here. E. All right. Monomer, active in allergies and parasitic infections, causes massive release of histamine. So that's going to cause a big inflammation response. Let's see what else it says here. Secreted by plasma cells in the skin, the mucosa of the gastrointestinal tract, respiratory tract, and tonsils. Not common in the blood plasma. Levels during severe allergic attacks and chronic parasitic infections of the gastrointestinal tract. Okay, so IgE still helps the worm being destroyed by binding to the worm, marking it for destruction by eosinophils. So if you have high levels of eosinophils, it's a worm infection. Eosinophils bind to the exposed stems of IgE. That's kind of convenient and easy to remember, isn't it? The eosinophil binds to IgE, which triggers the eosinophils to release toxic contents onto the prey, lysing it from the outside. So in other words, these worms are too big for the antibodies to take down themselves, but when they bind to the molecules on these worms, they cause the eosinophils to come in and release those granules that they have inside, the ones that stain red, and the contents of those granules will cause the worm to burst open when they're released, which is pretty badass and really ought to be in some sci-fi movie. All right, monoclonal antibodies as clinical and research tools. A monoclonal antibody is a weird thing. Okay, they're actually talking about here hybridomas. Good. That's where I was going to go. Commercially prepared pure antibodies that are specific for a single antigenic determinant. This is one of the things that's being used in immunotherapies against cancer. This is Rituxan is an example. This is amazing stuff. So we take a tumor cell and a B cell and combine them together to make a cell called a hybridoma. And because cancer cells are immortal, just keep dividing and dividing and dividing. These cancer-like hybrids that are made out of a cancer plus a plasma cell or plus a B cell, the same idea, they are going to be able to last forever and create more and more and more and more antibodies. And right there, it says tumor cells allow it to, the tumor half allows it to proliferate indefinitely, just keep going, keep going, keep going. While the B cell allows production of a single type of antibody. The hybridoma lets you make huge numbers of antibodies, which you can then harvest and use for therapy or for research. You inject those antibodies that you're making into the patient, and they will find the cells that need to be attacked and destroyed. All right, so antigen antibody complexes don't destroy antigens, but they prepare them for destruction by the innate defenses. The antibodies can go after extracellular pathogens, things outside the cell. The antibodies do not go inside solid tissue unless there's a tear or a rip. There was a recent exception. Antibodies can act intracellularly if they are attached to a virus before it enters a cell. What happens is they activate mechanisms that destroy the virus. All right, and this is the end of the second PowerPoint. So let's go ahead and stop that.